Yeah. Hi, good evening. Is my screen visible to you all? And can you hear me?
Yes, ma'am. Yes.
Okay, okay. Thank you. Thank you. Okay, so I shall begin. I think we are already on time. Okay. Mm. All right. Good evening. Thank you for joining. This is problem solving session 3. And like the previous two sessions, we will be going over the topics that you would have done in the third week of this lectures and this course. And these were the topics which were covered in the third week, which were more pertinent to muscles. So you would have started with muscles. What are muscle? How does a muscle look like? What is the structure? Then we moved on to skeletal muscle in detail, where we started with the formation of skeletal muscle. Then we looked at the anatomy. Just a second. Yeah, anatomy. And then we did something very important and interesting, which is the construction of muscles. When your muscle is contracting, how does it actually take place and what is exactly the mechanism behind it? And then in relation to that, we studied about the function of actin and myosin. So these were the 5 topics or 5 lectures that were covered in week 3. And today's questions will be related to these topics, which involve questions from the previous assignments and some new questions. Right. So I will just quickly go over the topics just as a refresher of what all did you cover it this week, and then we will begin the questions. So the questions will be on these topics. I have tried to keep them as how the lectures have been, but there might be slight up and down. So the first topic that the lectures began with was axial and appendicular skeleton. If you remember, axial skeleton was about basically skeletal portion covering skull, ossicle, which is basically the skull and the associated bones. Then the rib cage, the hyoid, which is the connection here, and then basically till the vertebral column. So the vertebral column and skull make up your axial skeleton, while everything else is appendicular, be it your limbs, be it your legs, be it your pelvic girdle, be it your shoulder, hands, anything. So appendicular means everything which is supporting the axial. Axial is the basic structure, the basic framework. So skull and the vertebral column, right? Now, after your skeleton, what we next did was just a second. Yeah, what we next is were type of muscle. If you remember, there were 3 types of muscles: cardiac muscle, skeletal muscle, and smooth muscle. On your slide, you also have the location where these muscles are found. So cardiac muscle, as the name gives away, it's in cardiac, basically heart. So it's the heart muscle. Then skeleton is basically all the muscles which are integral to skeleton. So the muscles here or above the bones. So skeletal muscle. And then you have smooth muscle, which are part of the internal organs that we know. Now, how do you differentiate between these muscles? First is striation. So your skeletal muscle has striations and cardiac muscles have striations. Uh, Talking about voluntary control of the muscles. So skeletal muscle, which is of course the ones which are connected to skeletons or bones, are voluntary. You can move them, you know when to move them, you know how to control them. However, cardiac muscles and the smooth muscles are involuntary. And then you have these nucleated and the how they are connected to each other. So they have intercalated discs. So cardiac muscles, characteristic feature is these intercalated discs. Rather, when you have this intercalated disc, this is cardiac muscle. That's a giveaway of the structure of cardiac muscle. So these 3 muscles are clearly differentiated with each other, and they have their own properties, right? So these are the types of muscles. But the main muscle that was discussed in the lecture was skeletal muscle. And one tip for you, whenever you are revising skeletal muscles or whenever you are revising muscles, especially skeletal, uh, be very careful of the terms that are used, because every term has its specific meaning. And my suggestion to you is just imagine it as a flow chart or a workflow, and that should be clear. So start from muscle, try to splice, or just to see a slice of the muscle, you will see there is epimysium, there is perimysium, so inside and then below perimysium. In those you have these fascicles, In each fascicles, you have several myofibrils. In each myofibril, which is one myofibril, you have several sarcolemma and sarcoplasm. In these, you have muscle fibers. In each muscle fibers, you have myosin and actin. So don't worry, we will be discussing this topic extensively today. There are a lot of questions that cover this topic. So I expect and I hope that at the end of this session or when you are revising later, these serve as important and helpful for you so that you can grasp the topic, right? So this is the basic structure of skeletal muscle, where you began first with understanding how are these forming. So you start with something called myoblast. They form myocyte, then they fuse to form myotube, which together again straighten up and align and form myofiber, right? So yeah, what I said is myoblast. So you have these myoblasts which fuse to form the myotubes, and you have these satellite cells which come onto these myotubes. Together, they form elongated structure, which is called the muscle fiber, right? They have each their important function, which we will be discussing in the upcoming slides, right? Now, from here, from this figure, let's go into a more detailed figure. So again, a bone. On the bone, you have the muscle. On the muscles, you have these different blood vessels. Each of these bundles that you see, they are called fascicles. In each fascicles, again, you will see say, several small, uh, you know, uh, units or components. Those are those are called basal lamina. So they're surrounded by basal lamina. You have sarcolemma, and the satellite cell that you just saw in the previous slide here is present inside this. So what we are seeing here is basically this. All of these, multiples of these together form up the muscles that we see, right? Okay, again, now why I have shown you this figure, because this one, the next one takes one step ahead from here. So you have understood till muscle fascicle, all these bundles are fascicles. Each fascicle has several fibers, so you have fiber. Now each fiber, if you open up and see, how does it look? It has several nucleus, yes, but it has several sarcolemma, which is the covering. Inside the sarcolemma, you have several myofibrils. Now, what is important in myofibril is this part, which I'm sure you would have done very distinctly if you remember myosin and actin and Z zone and M line, if you remember these. So let me show you this so that it clicks your mind and you are able to remember this. Do you remember sarcomere? You would have done sarcomere, and it's the most important thing in terms of muscle contraction. If you want to understand how muscle contraction is taking place, this is how it is taking place. This is how you should understand it. So let's look at the left one first to understand what is happening on the right one. So this is a sarcomere. Several of these sarcomeres are repeating in each other if you see here, right? So these are sarcomeres. All these sarcomeres together are forming up your myofibril fiber basically, okay? Now, in one sarcomere, let's just look at one sarcomere. How do you identify sarcomere from your fiber or myofibril? These zigzag lines. So the zigzag line mark the boundaries of sarcomere. Okay. And these zigzag lines are called Z line, right? So the Z line or the Z line marks the ending of the sarcomere. Start with that first, right? Now come zoom out and see what's happening. So if you take these portion and enlarge it, what you see is this is a zigzag line. And then in these zigzag lines, you have these kind of protrusions. The red ones in the center are the thick filament. What is a thick filament? Myosin. And the blue ones, which are here on the boundary of or from the edges, they are the thin filament, which is the actin. So you have actin and you have myosin. And then you have, for your understanding, you have several lines. So we started with Z line, which is the ending. One in the center is called the M line, or you can remember it as medial line. So M line. Okay, M line. Now you have two lines, Z lines and M lines. Okay, M lines is basically, so whenever you see the M lines, just know that it is the center of your sarcomere or center of your filaments together. So sarcomere. Okay, so that's the M line, Z line is the ending. There are two types of bands, A band and I band. Okay, it looks difficult. Once you understand it, it becomes very easy. Now, A band. See the boundary of the A band that is shown here, or the boundary that is shown here. What do you see as the start and the end point? The A band basically is the starting and the end point of a thick filament or myosin. Where is the myosin? The red ones. So you need to hold one end of myosin and the other end of myosin. That is basically your A band. Now, I told you to start from end and beginning. Now, when you do that, when you are actually looking at the A band, you are basically looking at the entire myosin and two actin filaments. So one myosin and two actin filaments. One myosin and two actin filaments. So you have actin filaments and myosin filaments overlap in the A band, right? So you can remember A as your first band or A as your actin plus myosin band. Okay. Now, what is I band? I band is the opposite. I band is basically consisting only of the thin line or the actin filament. And where do you find this? You find this here. So uh, between two ends of myosin, so here thick filament and thick filament, you have I band. So you basically you can remember it like this in terms of your arrangement. You have A band, I band, A band, I band, A band. And whenever you have A band, you have both actin and myosin. Whenever you have just I band, you have only actin. Now, and what is this H zone? H zone is the complementary to your actin I band, because in H zone, which is here, it's just the myosin or a thick filament. It looks very complicated, but once you draw it out or once you map it out, it becomes very easy for you to understand, right? So what do you see? You see A band, you see I band, you see H zone, M line, Z line. That's it. You should remember it uh, as the structure of sarcomere. And now on to the right one. The first one, which is the A1, is basically just what I have told you, right? The second one is basically when your contraction is taking place. So when your muscle is basically contracting or when your muscle is in use, you're basically getting all this big length of sarcomere in a smaller portion. So you're decreasing the length. As you're decreasing the length, look at the overlap between the bands. So you see this line here, Z line or the I band, it almost disappears. The Z line is very close now. You don't have this actin overlapping actin band at all. So what happens? This distance and this distance shortens. So these bands, these myosin come closer, or these actin and myosin come closer to each other and it shortens. So when that shortening takes place, that is your contraction. The name itself tells it's a muscle contraction. So it's contracting, it is getting smaller, and this is how it is taking place. Now, there is more mechanistic description to it, but before I go on and on about it, I thought we'll do questions so that you grasp the topic better. And we have this explanation of how this entire process takes place at the end. So hold on. We will do that also, but I'm sure by the time we reach that slide, there will be so many questions that you would already have had the revision of this part. So I'm sure that you would be able to do it there. Okay. So let's begin with some questions. So remember the 5 topics that I told that we are doing in this week or today's session. So the questions will follow in that order. Now, what are these 2 statements? You need to tell me if both the statements are true or if both the statements are false or if one of them is true or false. So basically, you just need to tell if the statements on your slides are true or false. So let me read the statements out for you. The statements, I think, are self-explainable. So smooth muscle contraction is slower but more sustained than skeletal muscle. Is that true or false? And the second one says tendons connect skeletal muscles to bone and transmit the force of contraction. Is that true or false? Let me know your comments in the chat box. I am seeing there. True. Both the statements are true.
Yes.
Okay. Anyone else? Others? Okay, true. I see two responses still now. Anyone else? Okay. So I see this, most of you writing true. Okay, let me give you the answers. And you are correct. So, and if you are typing and still saying this, yeah, I can true. Yeah. So basically, both the statements are correct. The first one says smooth muscle contraction is slower. That's because of lower ATPase activity of myosin. And if you know about ATPase activity of contraction, we'll do it anyway, but that's how your contraction is taking place. So in your smooth muscle, your there is lower ATPase activity of myosin heads, and it's more sustained because again because there is minimal ATP consumption, you can sustain it longer, and it is a slower process which is able to stay for long. And tendons connecting skeletal muscle to bone, something that we have done in the previous class already. Okay. Now, next set of simple questions. So, the first one is during muscle development, mesodermal progenitor cells differentiate into dash, and that later forms multinucleated myotubes. So, between your progenitor cells and myotubes, there is one step. What step is that? Next one is dash muscle fibers are striated, multinucleated, and under voluntary control, right? The one word is giveaway. Which answer is this? And the next one is the area between two Z lines in a myofibril is called a dash, and it shortens during muscle contraction. Very easy. These are technical terms, and that's because they are more like, what do I say? Descriptions. That's why I gave them here. But if you attempt them, this should be very easy. So one is about the skeletal muscle formation. Next question is about the muscle types. And the third question is about the major topic of this week, which is muscle contraction. Two muscle type. Yeah. So which muscle type? So so it talks about, right? This is the so second question. So is about, you need to answer me in terms of the three types of muscle. Which type of muscle has been spoken about? Characteristics are striated, multinucleated, and under voluntary control. So voluntary and striated. So the three options: skeletal, cardiac, and smooth muscle. Which option is this? Anyone? I'm sure the second one is easy, or at least take a guess on the second one. That should be easier for you to answer. Okay, one more minute and we can then discuss the answers, but keep guessing. It's okay. I mean, even if it's incorrect, it's okay. It's not a worry. Nobody? Okay, I see someone typing. Maybe I'll just wait for that person. Others can also chip in and at least, okay, maybe you can give me the first answer. Which answer do you think? Okay, second answer I've got. First answer, can you give me what is the cell called during muscle development? Basically, what is the second step after the progenitor cells? Anybody? If you remember distinctly the name also, it should be fine. Yeah, second one you have guessed already. The first one and the third one. The third one is basically, we discussed the unit right now, right? The Z line and M line and H zone. So what was the unit called? That unit is what I am referring to. Okay, let me show you and then I'm sure you knew it, you just didn't think enough. So the first one is, if you're looking into the muscle development, the progenitor cells first differentiate into myoblast, then it forms the myoblast fuse together and form myotube. So I remember these flow chart, okay? This is very, like you need to remember it in that order. Second, you already have guessed skeletal muscle. Now, let me tell you, whenever you see this kind of question or whenever you're trying to remember, if you see voluntary, just write skeletal. You don't even have to see anything else because only skeletal muscle is voluntary. Cardiac muscle and the smooth muscles, they are involuntary. Sorry, I just lost my connection. Okay, let me move to the next question before I again seem to lose my connection. Right. So we did the first topic, remember, axial and appendicular. This question will give you a revision of that question, that part of the syllabus. So axial skeleton, which are the food is part of that, and appendicular skeleton, which one is the part of that skeleton. And this will cover your one part of your lecture that week. What is axial and what is appendicular? If you remember that, this question is easy, and then that basically covers your topic. Remember what is axial? Axial is basically something that gives the backbone or access to your structure. And appendicular is basically anything that supports the main or the central, in this case, the central bone. So when you talk about axial skeleton, you're basically thinking about something which is giving structure to your body in this case. So which one would that be? And appendicular is anything which is not part of the axial. Okay, so that's first. What about the second one? One is C. Okay, what about the second one? Take your guess for about the axial and appendicular. Okay. Okay, let's look into the answers first. Right. So the first one, axial vertebra, and the second one is limbs and girdles, which is appendicular. Let me take you back to the first slide so that you can just quickly see once again. That's your axial and that's your appendicular. Once again, axial is basically that gives your structure. So your midpoint. So your vertebral column and your skull are axial. And everything else that you see, this is supporting this side, this is supporting this is supporting this. So you have your shoulders, your girdles, your pectoral girdle, pelvic girdle, limbs, hinds, the clavicle here, everything else is your appendicular. Just remember vertebra or vertebral column, which is basically also the rib cage in front and the vertebral column in the back. Hyoid bone, which is basically joining the skull and your vertebral column and your skull entirely. They are the axial and appendicular. They are the axial skeleton and everything else is appendicular. So when you come back here, your axial, femur, humerus are in leg and hand, so they're appendicular. Pelvis is again a girdle, so it's appendicular. Second question, appendicular, skull and ribs are the axial. Vertebra, again, the backbone, so your vertebra and the sacrum, which is at the end, which is your part of the axial, and sternum and clavicle, which is here. So again, your vertebra, the rib cage here. So that's sternum and clavicle. Those are appendicular. Those are axial. So limbs and girdles are appendicular. Okay. All right. Let's move to the next questions. These are on the type of muscles. And again, like I said, these are easy for you to remember. So first is non-striation. Second is involuntary. So you should now see striation. And then involuntary and striated. So one is non-striated, which one is striated, which one and involuntary. So which ones are the one? Okay. Right. C. What about the second one, which is involuntary and striated? So basically, you need to remember between cardiac and smooth. Okay. Anyone else? Okay. Let me move on. Right? Non-striated, smooth muscle only. Involuntary, smooth and cardiac, but striated means cardiac. Right? Okay. Next, Which muscle type can regenerate to some extent after injury? And the second one is cardiac muscle is unique because of which I told you this exact line. We identify cardiac muscle because of its feature, unique feature. And this one, try to think which muscle has this property of being able to regenerate. Okay. And A. Anyone else wants to take a guess? Okay. Others can also take a guess. You can think of any answer. It's okay if it doesn't be right. All right. Let me show you the answers because we have to go on time. The first one is smooth muscle. So smooth muscle can regenerate up to some extent. And the next one, which is cardiac muscle, is known because of intercalated disc. Remember, I told you it has this cavity-like structure in between that's called the intercalated discs. Okay. Okay. Now, uh, we already saw about heart, we already saw about muscles, and we already know about where smooth muscles are present. Now, in smooth muscle, you have something called multi-unit smooth muscle. So where do you find multi-unit smooth muscle in these options? It's okay, take your guess. It's the idea is just to give you information. So what do you think out of this, which one could possibly have multi-unit smooth muscle? C. Okay, anyone else wants to take any guess? A. Okay. Anyone else? A. Anybody? No? Okay. So the answer to this is A, ciliary muscle of eye, which is here. Let me tell you the others. What are the others? The others, all of them, uterus or gut lining or your bile duct viscera, they are all composed of single unit smooth muscle, right? And what is the difference between multi-unit smooth muscle and single unit smooth muscle? It is in terms of their ability to contract or signal transduction. So in terms of multi-unit smooth muscle, which is here in the eye, you have these different muscle fibers coming and innervating each other, right? And each of these muscle fibers or each of these fibers are innervated by a single nerve ending, right? So what you do is you have a control over your movement. So each of them are innervated by nerve endings, while the others, they work as a syncytium. So they work together. They work. This is a single unit, and they can work together. Like the entire unit works as a one. While eye has several muscles so that they can control the movement, which is important also because you're looking at different directions also, and you need different cells to work, right? Okay. This one is very easy, but it's just to end the topic. Uh, the first few topics. So a bone-to-bone junction is called, or in other term, what connects bone to bone? Yes. Anyone else? This is something that you should be like. This is something we have been discussing last time also. So this is easy. Yes, pretty much B. I see people are getting there. So ligament. Tendons and ligaments. Okay. Cartilage we discussed last time. Fascia we will see today. Okay. Moving to the next topic, which was about uh, your formation of muscles, skeletal muscle. So, first question is, which embryonic germ layer gives rise to skeletal muscle? Okay, anyone else? Take a guess. It's okay. Which one do you think did you read? Did one? This is also part of the films that we did in the first this thing slide. Anyone else wants to take a guess? All right. Since we have to move forward, it's mesoderm. So I don't know if it's in this time's course or not, but generally in developmental biology, you would often see these different germ layers give rise to different, like different region of your embryo. So there it is also, it becomes more clearer, but just because we are doing it topic wise, so skeletal muscle comes from mesoderm. Remember this. Okay. Now remember I told you we did about muscle formation. We told about satellite cells where I told you that myoblasts come and fuse and form myotubes. Then you have satellite muscles. Then they together differentiate to form muscle fiber and satellite cells. So I told you what are the satellite cells for. We'll do it in slides. So this is this question for that. So first you take your guess that which of the following function is the role of satellite cells, and then we can look at the answer. Which one of the following do you think satellite cells, which are there on the, like the muscle fibers, will do? You're free to take your guess. It's okay if it's wrong. Yeah, C, A. It's fine. Just type whatever you think is the most appropriate. C, C. So most of you are saying C, which is amazing because that's exactly the correct option. So satellite cells are generally used for muscle repair and regeneration. And remember, this is something that we just covered a few slides back, where we said that smooth muscles and cardiac muscles. We remember we did this question about regeneration. So the role of or the cell that take part in that role is satellite cells. Okay. Now, we did that which starts from mesoderm, which is the earliest precursor cell. So where does it start from? Where does this formation of skeletal muscle start from? You should remember what is sarcomere, so you know where it comes from. And what you are left with is myocyte, myoblast, and myotube. So which one do you think is the first type of cells that begin the formation of skeletal muscle? Is it myocyte or is it myoblast? Which one of that is the correct answer? Okay. Okay, so people have said A, B, A, B. Okay, let me show you the answer first. Myoblast. Okay. Um, we know myotube, we know sarcomere. Let's not deal with it because myotube is after myoblast because your myoblast will first fuse and that will form myotube. Sarcomere comes very later in step where you have the sarcomere and then you sarcolemma, all those things, right? Let's talk about myoblast and myocytes because I think people got confused here. And that's okay. So myoblast and myocyte, they are both related to muscles, of course, because the word MYO says that it is muscle related. Now, what comes at the beginning is myoblast. Okay. These myoblast and myocytes have a very, very clear distinction, and that's in the term of their commitment. What I mean, if you know stem cells and if you know how any cell differentiation takes place, you know that there are some cells that are non-committed, and then there are some cells which are committed. And then there are some cells which are mature. They will form this particular type of organ only. Let's come to that. So myoblast is the immature mononucleated precursor cells from mesoderm. We know mesoderm. From mesodermal progenitor cells, you have myoblasts, and then these myoblasts will divide and form. These are mononuclears, so they will fuse, they will form these multinucleated cells, which will form the myotube. Now, when do you call a cell myocyte? These myocytes are differentiated cells. They are these elongated muscle cells. Remember, at the end, you have this spindle-like structure. Those are myocytes. Sites means a clear, like we define it with region. So myocyte means at muscles, the cells which are present in muscles. Then we have adipocytes. There's some cells which are present in adipose tissue. So sites generally are defined with the region. So myocytes means they're already in the muscle. Myoblasts are cells that will give rise to muscle. So that's the difference between them. So one is differentiated, one is matured, and the other is immature. It's at the beginning. It will divide further to give rise to the cell type or the muscle type here. Okay. I think that's the last one here. So in muscle development, the structural protein that organizes these myofibrils. So basically, which proteins takes part in this structural component, which is a structural component of muscle units. Take a guess. It's okay if you don't know. See, some of the words you already know and you know where they are used. Do you think they will have a role here? For example, we have studied about keratin. We know about collagen. We also have a similar idea about elastin, and we have also have we seen about desmin somewhere. So think of their functionality and think if they are also they can have a role here. B. Okay, B, C. Okay. Anyone else wants to take a guess? You think it's between B and C? Yes, makes sense. If you would think about desmin and elastin. Anyone else? You know where's keratin, you know where's collagen. Do you think they have a role here as well? Similar role? Okay, no worries. So the answer is B, desmin. And you know where you find desmin? Where does it actually go and sit so that it helps the formation of the muscle or myofibril? It's basically the protein that helps align the Z line. What is Z line? Z line is basically the zigzag line between two myofibrils or between two sarcomeres. And what it does, it goes and binds, or it binds to the protein and it aligns the Z line. If you align the Z line, you are basically demarcating the boundary of sarcomere, and its help aligns the sarcomere. So that's how it helps in giving the structure to the muscle fibers. Okay. Okay. Now we know skeletal fibers are multinucleated. We have seen the images of multiple blue colored nuclei. So why do you think they are multinucleated? which is the best reason, which explains that why are these muscle cells multinucleated. Is it to provide enough genetic material for protein synthesis, or is it because nuclei have migrated from the surrounding connective tissue into the muscle fiber? Basically, these nucleus have moved into the muscle cells. Third, it is because of fusion of the multiple myoblasts. Or the last one is, uh, it basically ensures that all the sarcomeres or each of one of them have their own nucleus for control. It's an easy question. It basically highlights why do you get the final stage. Sure, sure. Yeah, please do. Please do. Please take care. Get well soon. Yes, C. Okay. Anyone else wants to take a guess? C. Others? Okay, this is one very smart crowd because they're thinking in this correct direction. Yes. So they are multinucleated because they are formed because of the fusions of different myoblasts. This we already know, right? And once they have fused and formed this multinucleated, they have this large cell now, one large cell with many nucleus, and this large cell has a lot of cytoplasm, right? And that can meet the production of protein. The nucleus doesn't ensure that. The high cytoplasm ensures that. So for the cytoplasm, you need multiple cells to come and fuse together. So that's why you are multinucleated, not the first one, not the second one, and definitely not the third one because you don't need nucleus for control. Very nice. Okay. Last one to give it in finishing. The fusion of myoblast gives rise to which structure, which I think now, by now, everyone is absolutely clear about this. So basically, what the question is asking is, after your myoblasts are basically forming, fusing, there is fusion of myoblasts together. What do you have? What the structure is called? No? Yes. See, I'm pretty sure you will all know it very well. Okay, myotube. We have done this quite a lot of time. So I hope you are able to visualize the entire process. So you have mesoderm, you have progenitor cells, you have myoblast, you have myotube, which then form the muscle fiber on the myofibril and the sarcomere ultimately. Okay, all right. Let's switch our gear and then move on to our next structure, which is muscle fascicle. Remember those elaborate structure where we saw muscle fascicle? What is muscle fascicle? What does the bundle actually mean? What does the bundle basically consist of? Okay. Anyone else? Okay. All right. So it's not individual muscle fascicles. I will show you the structure later again. It's the bundle of muscle fibers connected by connective tissue. So individual muscle fibers together are forming a bundle, and that bundle is called muscle fascicle. So that's the distinction that you need to make and remember. Okay. Um, yes. And let's then quickly and very easily jumped into the next section. So when you have a supercoiled assembly of actin and myosin filament inside the muscle fiber, so what is that arrangement called? Anybody? Okay. Yes. Sarcomere. Okay. Okay. Now the next question is about the bands. But before I go on to that, sorry. Yes, I see a lot of C's, so it is making sense. Now, before I go to the next question, let me quickly show you the structure once again so that we are all on the same page. So I will stop sharing for 2 seconds and I will come back again. Wait a minute. Hmm. So this will make more sense now because back then it would have confused you a lot. Is my screen visible?
Yeah.
Yes, I hope so. Thank you. Yes. All right. So here, what did we just do? So this was your muscle. These are your fascicles. Okay. So your muscles, when you see a slice of it, you will see a lot of muscle fascicle. Each of this bundle is fascicle. See, this bundle is fascicle. Now, if you see the fascicle, you will see a lot of muscle fibers. And that was the question. What is muscle fascicle? A bundle of multiple fiber. When you look into these fibers, see all of these fibers, when you zoom it like this, you see that it is consisting of many of these dot like structure, right? What are those dots? Those are all myofibrils. So together myofibrils form muscle fiber, and those muscle fibers together form a muscle fascicles. That's what the question is about. Now what we will see is when you go into those one myofibril, right? What does those myofibril look like? And then those myofibril, which is here and here, you have the sarcomeres, which are contiguously connected to each other. And we will see now how does those sarcomere, which are back to back, arranged back to back, how does those sarcomere work and how are they arranged? Okay. And that will give us an idea about the uh, how the contraction takes place. Okay, let's move ahead. Yeah. So now you know what is myofibril, the ultimate unit that we want to check. Now let's begin and try to understand all these different terms that we are reading. So in myofibrils, the band that contain myosin filament and overlapping ends with actin filament are called. Now my suggestion to you is, if you are confused, just try to remember or draw somewhere this structure that we have looked into, the sarcomere, and then try to answer these questions. This will become very easy for you. So I can give you some time. You can draw it out. You can map it out and think where is I band, where is H band, H zone, where is A band, M line, Z line, and all of these, which are the actin filaments, which are the myosin filaments. And after you have that structure ready, try to attempt this question and the following questions, which will assure that you have both understanding and as well as the visualization of sarcomere or myofibril for that matter. A band. Okay, others, please don't like rush into answering. You can draw, you can visualize, you can think, and then you can answer. So someone has answered A band. Do others agree? So I hope you know, or what is important for you to localize on your like in your sarcomere. For everyone who are still making or who are still visualizing, you should be able to know where is M line, where is Z line or Z line. You should know what is A band, what is I band, and what is H zone. Okay. Are you making or are you doing? Okay. What I will do is I will give the answer and then I have the image on the slide so you can compare. So A band, like someone mentioned, you were absolutely correct. Right. Your structures should look like this. If you are making B was, yeah, A band, exactly. So once again, let's look into this. Check if you were thinking right or if you're drawing right or if you imagined it right, right? Z discs are your beginning and end of your sarcomere. So one Z line to another Z line is sarcomere, right? Now you have thick bands, which are your myosin in the center, and the blue lines are thin filaments, which are actin. Okay. Your A band is basically your end to end of myosin. And if you look at the band, it will have actin, myosin both. While your I band will only have actin filaments, while your H zone will be the center. So your M line is the one at the center, and your H zone will have the center and only your myosin filaments. Okay, you should remember this. All right, let's try to make it more easier for you or more obvious for you. Right, let's go again. So the bands now that contain only actin filaments. Where was that band where I showed you only actin filament? I band. Anyone else? Need any other option or with the same option? Okay, let's move ahead. I band. So if you remember, I band here has only actin filaments. So I band. Okay. Now, the actin and myosin filament are connected side by side using which of these following proteins? Take a guess. Yeah, I band is correct. Take a guess. So you have your actin myosin, you know where they can come adjacent. So which of these proteins um, help in that connection? Again, at least two of these terms you know already. Do you think they have a role here or is it something else? Anyone? So it's titin or keratin, osteocytes or creatinine. I think you know the terms already. At least creatinine, osteocytes, keratin, you would have definitely heard. So do you think titin or any of these, I mean, out of these, which one has the role in this connection? A or C? Okay. Anyone else? Okay, let me give you the answer. It's titin. Okay, so this titin basically connects the myosin, thick filaments, and the actin, uh, and they are basically helping in the alignment of the sarcomere. And keratin, we all know, is found in the integumentary system, which is skin, hair, and nails. Osteocytes are basically bone cells, not muscles. And creatinine is another, it's a waste product of your muscle metabolism, not a structural protein. Titin is a structural protein. Okay. Okay. Now I said, remember, there was mechanism of how this contraction takes place. So you would have come across this term there, troponin. So let's just first discuss the questions, and then at the end of these MCQs, we will have the discussion on how does that mechanism take place. So you remember troponin. So you have to tell me which subunit of troponin had higher affinity of actin, and that will make you imagine what's exactly the mechanism. Okay. Anyone else? So if three troponins I, C, and T, anyone wants to take a guess? All right, let's give the answer. So it's troponin I. So if you remember, troponin I, it basically it binds to the actin filaments, and then when it is binding to the actin filament, it inhibits the actin and myosin interaction. Okay? And this happens when the myosin is relaxed. So when the myosin is relaxed, the actin is bound by troponin I. And because it is binding to troponin I, or because it is bound to actin, actin cannot interact with myosin. Okay, so that's why troponin I. Troponin T, it binds to tropomyosin, which is on the myosin. Okay, and troponin C, it basically is for the calcium ion, so it helps in the contraction. So out of these three, I is for actin. Okay, yeah. Right, correct. All right, next one. So now muscle fiber we know consists of myofibril. And then each myofibril consists of certain number of actin and certain number of myosin. Now imagine whenever you see actin and myosin, what is the relation of the number of filaments of these different, like in a sarcomere? If you look into the number of actin and filaments and actin myosin filaments, how are their numbers related? So question is, you should see it in perspective of actin is to myosin. So how many actin and how many myosin in one fibril, myofibril? Double. Anyone else? So you should see in terms of equal number, double, half, or one fourth. So will actin and myosin will be equal in number, or do you think actin will be more and myosin will be less, or do you think it will be opposite that myosin is more and actin is less? No, anyone? Okay, let me show you. So it's double. How it is double? Very simply, in each of these myofibril, if you see this myofibril, you have this one myosin as your, if you take this myosin as your reference, then you have 1 and 2 actin. So twice the actin. Again, if you have one myosin here, you have 2 actin overlapping. So one myosin, 2 actin, one myosin, 2 actin. So it's double. So it's double the number. Okay. All right. Very easy. So when there is an actin myosin overlap, what happens to the sarcomere length? Or in other terms, when there is muscle contraction, what happens to the sarcomere length? It's very simple. So if a muscle is contracting or if the sarcomere is contracting, what do you think will happen? Okay. Uh, Someone is typing. Let the person type. Only then it will increase. Don't you think it will decrease? Because when we will see when there is actin myosin overlap, it's basically pulling the sarcomere or pulling the actin myosin filaments closer. So your length is shortening. Okay. If you don't remember, I don't think so. I have the image here. Maybe I can show it here. So when you have actin and myosin getting closer, they will pull each other. Remember, there was a side eye image I showed you that these all became very close. Your eye zone disappeared completely. So your filaments or your sarcomere fibril length will overall reduce not increase. Increasing means getting outside. It will come closer to each other, so it will decrease. So remember that. It's the name itself says, right? Muscle contraction. So it is contracting. Okay. All right. There were some terms which were extensively discussed in the lecture, so I'm just going to cover them in one question. So if so this question says, if load applied on skeletal muscle equals maximal force that can be exerted by muscle, velocity of the contraction becomes dash, and even when muscle fiber remains this. So try to remember that topic and try to answer. It's okay if you don't. I will wait for 1 minute and then I will give you the answer. Do you remember something about load? Do you remember something about the graph that he was talking about? So let's try to understand and remember that. C. Okay. Anyone else wants to take any more guess? D. Anyone else? Okay. Let me give you the answer. So it's 0 and activated. Now, what does it mean? So what does it say? If load applied on the skeletal muscle, which is basically the force, the tension, equals the maximum force that can be exerted by muscle. So it means that the muscle is something which is in which, so the muscle, each muscle can have a certain capacity to generate force. Only with certain force can you work. So if that maximum force generating capacity is reached because of the tension that is applied on the muscle, then what happens is, so you are, how do I say? So your contraction is what is driving your shortening of your sarcomere only. We have just read, right? So you contract your muscle to do some work and then that's shortening the sarcomere, right? Now, when you have reached your, how do I say? You have reached your maximum capability of force generation. You cannot contract anymore. So when there is maximum force that has been exerted by the muscle already, there is no more contraction that can take place. So zero contraction, even if all the other processes which we just read about overlap of actin myosin and that there is overlap, you will see there is removal of ATP, there is removal of inorganic phosphate, ATP generation, everything else can take place. But because it is at its maximal length and maximal force capacity, it will be zero contraction. So that's the idea here, that when you have the maximal load, which is the maximum force that can be applied. Just imagine it, anything at maximum means we say, right, the muscles are exhausted, the muscles are exertion, we are exerting the muscle. That means we have reached the maximum capacity. It cannot contract anymore. Even if you keep thinking that it will generate more ATP and it will use more energy, it will still not, it will still not show any effect. Okay. All right. And this is something which is related to fast twitch and slow twitch, if you remember. So that's a topic that you should also check. So myoglobins are present in which kind of muscles? Remember, fast muscles, slow muscles, fast twitch and slow twitch. So you just need to remember this from that perspective. Where do you see more of myoglobin? Which kind of muscles? Remember the twitches, fast twitch, slow twitch muscles. You remember these are in relation to people sprinting, people running, people jogging, who use their muscle more for activities. So there we saw that there is a difference between the myoglobin content. There is a certain type of muscles which are, or there is certain uh, muscles which respond more to fast twitches, slow twitches based on their usage. Okay, let me give you the answer. So myoglobin is mostly in present in slow cells. Now, what are these slow cells or slow twitch? These are type 1 muscle fibers. Remember it like this. So these slow twitch, they are the muscles which are used extensively for aerobic activity. And if you have more aerobic activity, that can only be sustained because of the presence of myoglobin. So your myoglobin is present in the slow muscles. These slow muscles are red colored because of the myoglobin, and these myoglobin store oxygen because you need them when you are using them for your aerobic activities. While the ones which are fast muscles or fast twitchers, they require less myoglobin because they don't work under that much aerobic pressure. They are more generally relying on anaerobic metabolism, and that's why you don't have much myoglobin there. So remember, these two topics I haven't discussed in much, but you should check once when you are revising, or try to remember these topics as well. Load and fast and slow twitches. Okay. Okay, easy questions. These are very simple questions. So which ion is critical for muscle contraction by binding to troponin? Yeah. So NaNSE. Okay, so let me give you the answers because we are short on time. Calcium. So we know that. Also, we have seen this in one of the troponin question as well, right? Remember troponin ICT that we discussed there. Okay, next one. Now, which statement best describes actin and myosin during muscle contraction? So the muscle myosin slide past actin. Actin binds calcium to start contraction. Myosin head pull actin filament, shortening the sarcomere, or actin breaks down ATP for energy. So what is exactly driving the contraction? Hmm. Anyone else? Anyone else wants to take a guess? Which one is the answer for this? What is exactly happening when you have muscle contraction? Okay, let me give you the answer since we are short on time. So when you have muscle contraction, you have the myosin, which the myosin head will pull the actin filaments closer to each other, and that will shorten the sarcomere. The theory name here, if you remember from your discussion, it is sliding filament theory. So that's the theory that governs this entire process. Okay. Okay. Now, we already did calcium, which is about troponin. Now here, which ion is primarily responsible for depolarization in muscle action potential? This is something that is, generally discussed as well. So if you have a muscle contraction, which of these do you think has more role in depolarization? See, anyone else has any other option? Okay. So the answer is sodium. So in general, also when we are talking about action potentials, you'll see sodium and potassium have a very high, very great role. And for your muscle action potential, you will see that sodium has a role in depolarization for. Okay. Now, which protein regulates actin binding site and blocks myosin access at rest? Anyone has any question? See? Okay. It's actually tropomyosin. So you will see when we will see the reactions that so tropomyosin regulate the binding sites, and so it's basically on myosin and it blocks the access to the actin. We will see in a while. Give me some time. Yeah. Right. This is the easiest question probably. Thick filaments are made up of? Very easy. So thin filaments and thick filaments, actin and myosin, which one is what? Do you want to answer as well? I've got one response. Do you think it's myosin or actin? Yes. See, I told you where people are very smart. So it's myosin. Okay, quickly again. So now think and answer. When your muscle is contracting, which of these is actually shortening? We have discussed this. Just imagine which one is the one which actually disappears or shortens. Okay, one answer. Do you also want to answer? You can take a guess thinking what of you feel will be shortening. Mm, okay, no pressure. I will give you the answer and you can check it again. So I band, remember the I band is the only actin, the band which contains only actin, right? Now, if your actin and myosin are coming close to each other, this actin will move. So your I band will basically shorten up. So in muscle contraction, your I band is shortening up. Remember it like this. Okay. Again, where is H zone located? Think and tell, where is H zone located? End of actin filaments, center of A band, thick and thin filament overlap, or at the Z, at Z disc or Z line. So that I have given all these questions because this question, even though they are asking the same question, they're using different words and it makes you think about the topic more. So it really helps when you have already done these exercise, it comes handy when you are giving exams. Okay. Okay, I will give you the answer. So because we are only 28 minutes into the left for class, I will give only 1 minute to each slide and we will move ahead quickly. What the idea here is that you look into the question and try to solve it. If you are not able to do it, I will give you the cues for understanding and why is it like this, so that when you are revising, this comes handy. Okay, I'm going to give you all the set of questions that are there. So this one is H zone. So Z line, if you remember, Z line is the extreme end of the sarcomere, which is not what we are considering. H zone is at the center, M line, right? Remember M line and the up portion to the left and the right. So that's the center of A band, and H zone has only myosin. Okay, next one. Now, think in terms of contraction, and this H zone we have just discussed. If your muscle is undergoing contraction, what will happen to H zone? I is shortening. What happens to H zone, the one in the center? Will it stay there? Will it disappear? Will it shorten? Will it increase? Do you want to attempt as well? Do you want to take a guess? You can take a guess. There is no harm in taking a guess. Let me come to the answer. So it disappears because if you see, H zone is basically the filaments. So if you come back here, it's only myosin. Now your myosin and actin are coming close to each other. So there's no only myosin left. So your H zone disappears. Okay. Now M line. So it's a center line we know. What does it do? So does it anchor thin line? Does it mark the boundary of sarcomere? Does it connect adjacent Z disc? Or does it enter the central thick filament? No? Okay. So it is the, remember M line is in the center and it is the center of the myosin filament. Okay. Okay. Now this is the other term that I did not talk. See, there are so many topics on this lecture that I couldn't cover in detail, but I gave a question related to this so that you are aware. So there was something called power stroke. So do you know what is power stroke? Can you recollect what is power stroke? Or when do we use the term power stroke? Um, Anyone else? Okay, 1 minute over. So it's the same thing, that sliding filament theory or contraction, it's the same thing. When that contraction is taking place, it's called the power stroke. So your myosin head pivoting and pulling the actin filament is called the power stroke. Next, the zone of optimal interaction means what? I will wait for 1 minute and then we can look in the answer. In the meanwhile, chat box is open. Please, whatever you feel, if you know the answer, please put in there. Don't feel hesitant. Okay. So zone of optimal interaction refers to maximum overlap between actin and myosin filaments for force generation, right? So when you are in the process of force generation, the overlap between the actin and myosin, the region before contraction, is called the max or zone of optimal interaction. Okay. Okay. So again, coming back to then, in resting muscle, when muscle is not contracting, tropomyosin, where is the tropomyosin present? Is this very important? This question will these are steps that you should remember. So this question should help you understand the first few steps. Okay, so the answer to this is calcium binding sites on troponin. So we will see that your resting muscle, tropomyosin is present. So it basically blocks the calcium binding sites on troponin because they are interacting with each other. Next, according to sliding filament theory, which is what we have been discussing so far, muscle contraction occurs as what? Like what happens exactly in the muscle contraction? Okay, so let's quickly go over it. So your actin filaments slide over myosin and it shortens the sarcomere. Remember these sliding filaments theory and telling what exactly is sliding over the other. Okay, and this is probably the last slide. Yes, for this part. So the energy for myosin power stroke or this movement of actin and myosin, where does this energy come from? It comes from B. Yes, exactly. So your energy basically is driven by hydrolysis of ATP. And where does that hydrolysis start from? It's basically the energy which is in the myosin head, right? We remember the myosin head pivots. So that pivoting is because of the ATP hydrolysis. And that ATP hydrolysis then drives the pulling of the actin and myosin, right? So what I wanted to tell you is it looks like a lot of questions, but when you are actually trying to understand the topic, these are the questions that it keeps coming up. And what my intention was to give you all these questions so that you can practice when you are revising, right? So I will now show you what's exactly happening when your uh, contraction is taking place. Okay, so see here. The first thing is your myosin. This is a thin filament, thick filament. This is a thin filament. Okay, red and blue. This is your myosin head. They look like this, something like this. Okay. And in this myosin head, you have ADP and ATP. So you will now see hydrolysis. Okay. So the first, your myosin head attached to the actin of myofilament, and this is called cross bridging because they are like this, right? So this reverses, flips, and attaches to the actin here. So that's forming a cross bridge. Now, after this has happened, so you have then inorganic phosphate, which is being generated. How is it being generated? You're basically hydrolyzing ATP, right? So you are inorganic phosphate, which is generated in the previous cycle, that is released. And this initial power stroke, so it begins the power stroke. The myosin head will pivot. So you see, myosin head was like this. It will now turn, pivot, and attach and bend, and it will pull the actin filament towards the M line. The M line is here. So it will pull the M line. You want to see here, it is basically pulling here inside. So it's pulling towards the M line and the ADP is released, right? Now, after pulling, what happens? Now, when you have this, as the new ATP attaches to that, then your ATP will attach the myosin. This linkage will break open and they will go back to their original step, right? So you will first connect here, bend here, turn it inside, and then ATP will join and it will break open. Now you see you have tropomine and tropomyosin, which are all interacting partners, and we have seen through several questions that to make sure that the muscle is not always hyperactive and not always pulling, you have this troponin and your tropomyosin, which are blocking these interacting sites. Only when it needs to pull, you have this ATP hydrolysis and all this troponin go away. There is calcium release and it begins to, pull the actin to each other. So you see here, so all these, this is relaxed. And when you have, see, this is what we meant. See the M line and then see the red lines here or the actin filaments. And then you see the contraction, you see the M line, and you see how close is actin has now come to each other. So your H zone has completely disappeared. And you have a lot of calciums which were earlier binding onto the surfaces have now all been released. So they can all bind with each other. When you don't read contraction anymore, all these cross-linking will break open and they will all go back to relaxed stage. That's what is the whole idea about sliding filament theory. Okay. Now, in the next 15 minutes, we will just do these kind of questions that will help us understand these questions or these topics more. Right? We have done this question and this basically makes you think more. Okay. Now, the first one here is the statement says heavy meromycin contains, so it's basically your myosin. So your heavy meromycin is basically these two statements. Okay. So heavy meromycin contains the ATPase activity required for muscle contraction. The light meromycin forms the tail region of the myosin molecule. So do you think these two statements are correct and in relation to each other? So you can take, I would like you to attempt these answers so that we have time for discussions also. So please do attempt and see if you find any difficulty. Okay, anyone? Would you like to answer as well? Do you think the statements are correct and they are explaining the first one, like the second one is explaining the first one? Feel free to answer. Even if it's wrong, it's okay. Okay, let me give you the answer for this. So these both are correct statements. And so your heavy meromycin, it can do the ATPase activity on its head, because light meromycin, which is the other chain, it is forming the tail. So head and tail role is clearly differentiated here. Okay. Okay. Now, next one says troponin C binds calcium to initiate contraction. Okay. And calcium binding to troponin causes tropomyosin to move and expose actin sites. Read this and say if this is correct. And if this is correct, we will relate it back to the figure that we have just seen. Take a guess whether it's correct or wrong. Okay, let me give you the answer for this. So this is correct. So now see, remember this, okay? Your tropomyosin is here, okay? Now, it said that your troponin C binds to calcium to initiate construction. And as your troponin binds to calcium, your tropomyosin moves and exposes actin sites. Now see, your tropomyosin was here. If you see the tropomyosin, these are the tropomyosins, okay? Now as your troponin and your calcium is binding with each other, they are causing this tropomyosin to go away. And as the tropomyosin go away, actin and myosin come in contact with each other and they can interact. So they can start the contraction. So what it means is initially tropomyosin was hiding the actin sites, and that's why actin and myosin couldn't interact and you don't have contraction anymore. So that's what it meant. Okay. Okay. Next, skeletal and cardiac muscles are striated because of the arrangement of actin and myosin filament. And striations arise from the regular repeating pattern of sarcomere. So basically the striations, where are they coming from? Remember now, does it make it easier for you to understand that you have this muscle, you have a lot of striations. Are these striations because of sarcomeres? And are these striated, or are these um, variations in color because of myosin and actin? What do you think? There's a reason why myosin and actin are called thick and thin filaments. If you put thick and thin with each other, they will form some gradation of color. It will make you see like a striation. And that's exactly why you see striations in these muscles. Okay, so now you know striations also has a definite reason. Okay, next one. Myosin filament have heads that bind to actin during contraction. Myosin heads form cross bridges with actin and pull them inward. So this we know is correct because we have been discussing this so far, that your myosin and actin bind and they pull it inside. Okay, next one. Now the sarcolemma becomes depolarized when an action potential arrives and voltage gated sodium channel open, which is something that we have discussed. Okay, the answer to this is A, because remember we talked about sodium ions being responsible for action potential, and that's also why you have action potential in muscle, and that's taking place in sarcolemma. Remember I told you sodium, potassium, they take part in such uh, action potentials. Last or probably second last. So the A band correspond to the full length of thick filaments. The A band remain the same during contraction. This is important, and do you think this is correct? Okay, so this is again correct. Now remember why it is same in length. This is your actin filament, and A band is basically the actin filament. If you want to say that A band is shortening, it means somehow your actin filament, or sorry, I mean myosin filament, your myosin filament is shortening. That is not what is happening. You don't have shortening of the filament length. It is not that during contraction your myosin suddenly will become smaller. Your myosin will still stay myosin. So your A band actually remains intact. It's the I band, it's the actin moving closer. So remember this central band is stationary. Your actin filaments are coming closer, so your other lines and bands are shortening. A band doesn't move because A bands moving means your uh, myosin filament will shorten in length, and that doesn't take place. And then contrary to this, Z line anchors the ends of actin filament, and Z lines are found at the center of A band. Do you think that is correct? So this we know is not correct because A band is at the center and actin filaments is at the extreme end. So Z lines are not at the center of A band. Okay. Now I will end the session today with three questions which are very interesting, and I understand that this will be too much for us to understand and think of. So what I will do is I will give you the question, give 1 minute for you to absorb it, and then I will give you the answer so that you can understand what the question says. Okay, you don't have to necessarily think, but it would be beneficial for you when you think of what is the application in real life of these topics. Okay, the first one. A sprinter trains for long distance running. Okay. After several months, which of the following changes is most likely to be seen in their skeletal muscle? And this is related to your twitches. If you remember, we learned about twitches and we saw that how a person practices defines what kind of muscle intensity that person will have. So I hope you understood the question. Again, you have a sprinter who's training for long distance running. So basically, he's running every day very fast. So, what kind of changes do you see in skeletal muscle of these kind of trainers? So, the options are you are seeing a decrease in the mitochondrial density and have an increase in the glycolytic enzyme activity. You see a change in the myoglobin content and the mitochondrial density. You see an increase in the fiber diameter of the muscle and reduced oxidative capacity and decrease in capillary supply and ATP production by oxidative photophosphorylation. Well, the answer, as we can guess, is about the myoglobin content because we already read that when you have a slow twitch muscles, you need more myoglobin. So these long distance are also called endurance training. And the characteristic of these twitches are that they move the muscle to oxidative metabolism. So your mitochondrial density will increase, your myoglobin will increase, your capillaries will increase so that you can use more oxygen, right? While the first one here, which says decrease in metabolic conditions or mitochondrial density and glycolytic enzyme, those are for anaerobic conditions. Okay. And so because it's a long distance running, these are basically endurance. So they are endurance, they are basically for endurance training. This is for sprint training. Okay. C is again strength training and D is opposite of endurance training. So there are two types of training, endurance training and strength training. This is from that. Okay. Uh, we will see the professor extensively discuss this. Next type of question is, a drug stimulates chondrocyte proliferation but inhibits vascularization. What is the most likely outcome? Okay. So if you remember, this is from the previous session. So basically, we are saying that you have chondrocyte proliferation, but you don't have blood vessels coming in. So what will it happen? It will have enterochronal ossification stunted. So remember last time we did types of bone ossification. So if you don't have vascularization, but you have chondrocyte proliferation, it means the proper ossification will not take place. And the last one, which is again from one more topic that we have read, a person moves from a cool, cloudy climate to a hot, high UV region. So what happens? Sunlight increases. And which of the following adaptation is least likely to occur over time? You will see reduced dermal vascularization to conserve it because there is what will happen. First thing is increased melanin production, of course, because it needs protection from sun. Increased sweat gland activity because it will have sudden exposure to heat, so it needs sweat gland activity, and thicker dermis to with more collagen cross-linking so that it can protect the skin, right? So the last option where you have vascularization to conserve it, you don't need to conserve it because you now need to give away the heat. So what I wanted with this last three questions, with these questions, if you see, are from the topics of the past three lectures, today's and the two before. These are the type of questions that connect these topics to the like real world, and this is what the professor of this subject started with as well, right? So this is just a way to show how these questions connect you to the real world situations, right? Okay, that brings an end to the session, which is full 2 hours today. Yeah, so if you have any doubt for any of the three sessions, please post it. Otherwise we see you in the next class. Thank you.